Thank you so much. Thank you to the organizers for this opportunity. My name is Ayse, and I'll start with taking a quick step back in time. When we first sequenced the human genome, it took us over a decade and about $3 billion. This first generation of sequencers were highly accurate, but painfully slow and expensive. Then around 2005 came a revolution. This is the next generation sequencing, or NGS for short, and these platforms were able to sequence millions of DNA fragments in parallel at a fraction of cost and time, significantly democratizing genetics. And this transformation was also evident in CMT research. Especially after the commercialization of exome sequencing, we saw a boom in gene discovery in CMT research. Between 2010 and 20, almost 70 new CMT genes were discovered, and most of these were done using NGS research. It was truly a golden era. But today, if you read any cohort studies in CMT, you'll also see a pattern. We seem to be unable to provide genetic diagnosis to 30 to 40% of patients that are clinically diagnosed, but that they do not still have a genetic result. That is a lot of people looking for answers. Part of this diagnostic gap is obviously biological, but there's also a technical reason that I want to talk to you about. NGS reads DNA through short fragments, typically 150 base pairs long. This causes challenges in mappability of these regions, in especially regions of high homology or low complexity. This is where long reads come into the frame. These new platforms can sequence tens or even hundreds of thousands of bases long, providing us a continuous overview of the genome. It is almost switching from reading torn-up sentences in a book to reading entire paragraphs. Suddenly, the complex context becomes clear. In the next few minutes, I'll tell you how we use long-read sequencing to improve genetic diagnostics in CMT disease. But I also have another motive. This technology is quite new, and we now have the chance to make it more open, more collaborative, and more globally inclusive. That's why I want to tell you not only what worked for us, but also what didn't, so that others can move forward further and faster than I did. In this study, we used nanopore sequencing in a cohort of 35 CMT individuals from 14 families. Our patients come from genetically heterogeneous populations, such as those with Turkish ancestry, Bulgarian Roma populations, or patients from Brazil. Our entire bioinformatic pipeline and analysis strategy is laid out here, but I cannot go over all the details. But if you're interested, please come talk to me afterwards, and I'll be very happy to help you. Using this approach, we achieved long enough sequences and a pretty good coverage of genome in most of the sequenced individuals. But more importantly, we provide a 28% diagnostic uplift in this cohort. That is more than 1 in 4 families reaching a destination in their diagnostic odyssey. I'll give you a few examples. This individual here had a de novo variant in ATP1A1. But if you look at the exome data of this patient, this region is only covered by 2 reads, neither capturing the variant. On the other hand, if you look at the nanopore data of this patient, this heterozygous variant becomes very clear. This patient, who was referred to a research laboratory almost 15 years ago, received a genetic diagnosis now only because of long-read sequencing. The next example is a nanopore sequencing from a single individual, where the purple blocks here show a big insertion, more than 1,000 base pairs long. And this is actually the homozygous pathogenic GAA repeat expansion in the frataxin gene, which we saw yesterday can mimic CMT disease from Tanya's talk. This, again, was not captured in the exome sequencing of this patient because this is a deep intronic variant, and those are invisible to exome data. And as promised, I'll also tell you about the challenges. We map our DNA sequences to HG38 reference genome because that's the current golden standard. But currently, a complete telomere to telomere sequence of the human genome is available, and people will soon switch from HG38 to T2T genomes. We also experimented with this build, but honestly, it did not help me much, and I'll show you why. In this sequence, for instance, I think there might be a variant in this region, but this sequence on chromosome 9 is a completely new sequence that wasn't previously there in HG38, but is only present now at the T2T sequence. And also, this empty space here actually tells me there are no gene annotations available for this sequence, which basically means I don't even know if there's a gene or a gene regulatory element in this sequence that this potential variant might exert a functional impact on. So it's quite difficult to interpret this variant. If it's pathogenic or not, we have no idea. Therefore, I will currently stick to HG38 for another few years, I would say, until gene annotations for T2T is complete. Then, variant calling is a very dynamic field, and my only advice to you would be to not get too attached to one single pipeline, but implement improved tools as they come. Structural variant merging, on the other hand, is a very messy subject. In this example, it's kind of clear that this individual has a variant in this locus, but if you look at this person's similarly affected cousins, the mapper kind of gets confused and starts ambiguously mapping these variants into different genomic coordinates, and that's because this is a repetitive sequence. And if you try to call for shared variants among these three individuals, you'll fail, even though this is the genetic culprit of the disease in this pedigree. So I would also suggest you to be careful if you're sequencing multiple affected individuals and trying to call for shared structural variants. Variant filtering is the geneticist's detective work, sifting through hundreds or thousands of variants to find the one culprit variant is no easy task. But for SNPs and small indels, we have a lot of experience with NGS, so they're kind of routine. On the other hand, structural variant interpretation is still quite challenging, and that's because we can't even tell if the variants that we see are rare or not. And that's because the large population databases are built on short-read data, and those are significantly underrepresenting structural variation in the human genome. In this example, for instance, I was very excited because I thought I found the new CMT-causing gene, because my patient had this very impactful variant in a very interesting gene called KIF25, and I looked at Nomad SV database to see if this is a frequent event or not. And in this locus, there wasn't a structural variant reported that is similar to my patients. At the time in the department, we had about 275 long-read sequenced individuals available. So I also cross-checked to see if this variant was called in any of them. Unfortunately, though, almost all the individuals with the long-read sequencing data had this variant. So it wasn't a rare event at all. It was just invisible to short reads, but only visible through long-read data. So I have also many other challenges that I can tell you, but I guess I will not have time, so I'll just go to my conclusions. But again, if you are interested in any way, please come talk to me afterwards. So NGS revolutionized genetics and CMT genetics, but there's still a diagnostic gap present. Long-read sequencing offers a new paradigm now, and it can call for variants that are missed by short-read sequencers. In this study, we achieved a significant diagnostic uplift in previously unresolved CMT families, but also faced major analytical and interpretive challenges. Still, though, I believe long-read sequencing will move from a niche research tool to a first-tier diagnostic test very soon, especially for clinically heterogeneous diseases such as CMT. And making that shift, accelerating that shift is only possible if we share our experiences, tools, and data openly. That way, we won't be making the same mistakes in parallel, and we will be helping the patients receive their diagnosis. With that, I'd like to thank all the affected members that I have talked about and the referring clinicians and our collaborators. I'd like to thank my team and the sequencing experts at our facility, at our center. And thank you for your time and attention.
Questions in the audience? May I have one question regarding, it's more technical, but I think it's important, even happened with exome and genome short-read sequencing. I mean, for the data you showed here, I mean, I can imagine that the coverage is pretty good, but what about the depth in lecture? Because you have 2 patients with only 11 per.
Yes.
And there could be a problem in a specific patient.
Exactly. So coverage, we generally get quite good coverage in the 20 to 30x. Sometimes we also go all the way up to 50x. But we have 2 individuals with less than 20 coverage, 20x coverage, and that also definitely creates a bit of a problem. Especially though those patients were archived samples that have been sitting in our fridge for more than 10 years or so. And this is also evident in the sequencing data. We don't seem to be getting enough coverage with those archived samples. This means this is actually another example why we should engage patients as active research partners, because we will need fresh DNA samples to increase the coverage.
Okay. I can imagine that duplication and deletion, I mean 17 chromosome duplication and deletion is very easy to diagnose with using Lorenz scan, of course. Any other questions in the audience? There is Honduria over there, just in the corridor.
Hi. The long-read sequencing, it's, as you said, quite niche so far, and especially in underdeveloped countries, it will not be readily implemented. What do you think of any efforts to perform, let's say, linkage analysis to new variants so that you could actually use the old NGS sequences to maybe get some idea as to which variants you might be missing? Because there will be some rare SNVs that you will find in the area that you can link to your mutation. That way, if you can make a data set out of that, people can see if the variant might be present or not.
Well, yes, if you can catch those patients that already have like an informed loci, it will be easier to do a targeted analysis of those regions without going to long reads. But if you have no idea what's going on, if you have mimics of CMT, for instance, that you have to see a clinic agnostic view of the genome, then you will need long read sequencing, and that is the difficult part, especially with under-resourced countries. It is a very expensive technique.
But if you would already know which SNVs are there, like because they will do NGS sequencing data, they can maybe check for SNVs and then see maybe these variants that we know we will be missing with this technique can be present.
Maybe, yes. Some variants will be available through different tools that use NGS data to call for structural variants, it's true.